How do genes in our cells instruct the body to work? Cells are the basic building blocks of all living things. Our body alone is composed of many different types of cells that form the various tissues and organs and carry out specialized functions essential for life. Cells are made up of many parts, like a house with many rooms, each with a different function. The nucleus of the cell serves as the cell's command center. Let's imagine a nucleus as the kitchen of the house. The nucleus hosts our genetic material, called DNA, which, just like a cookbook, contains many recipes, our genes. Thousands of different genes can be found in our DNA, and these will guide and instruct the cell on how to make proteins. Different proteins can be imagined as different dishes or meals. Proteins do most of the work in the cells. Sometimes there may be an error in the recipe to make a specific protein. The protein coming from this incorrect recipe will likely not work properly. These errors are usually called genetic mutations and can cause medical conditions known as genetic disorders. What is gene therapy and how does it work? Gene therapy is a technology that has the potential to treat or prevent the progression of genetic disorders. Depending on the problem that is present, scientists can use gene therapy to remove and/or replace faulty elements of a gene. This is called gene editing. Prevent the production of a specific protein. This is called gene silencing. Transfer a full copy of a gene to correct a faulty or missing one. This is called gene transfer. In order to transfer genes or replace incorrect ones, gene therapy uses a vehicle called vector. The vector functions very much like a delivery package with an address that will deliver a correct recipe for a specific gene or transgene right into our house. The package will be delivered to the kitchen of the house, or nucleus, to produce a protein of interest, a protein our body needs that is either incorrect or missing. We can think of viruses as natural vectors, as they have the ability to transfer genes when they infect people. Scientists use different types of viruses as vectors, but these are modified so they can't cause disease when used in people. Adeno-associated viruses, AAVs, are among the most promising vectors to be used in gene therapy because they are relatively harmless viruses. AAVs are usually very small. Therefore, they have a limited packaging capacity, meaning that AAVs can only contain and therefore deliver relatively small genes. This characteristic may limit the use of AAVs in genetic disorders that require the transfer and the correction of large genes. AAVs can only contain recipes or genes that are no longer than approximately 5,000 characters or letters. Thus, great efforts have been directed towards identifying strategies to overcome this limitation. Shortening the length of the recipe while keeping all the most important information of the original recipe is one of the approaches commonly used. Let's imagine that the gene we want to correct, replace, or transfer is responsible for the production of a cake in our cells. We would not need just the recipe and ingredients to bake the cake, would we? For example, we would need to turn on the oven in the kitchen and set it to the right temperature. Gene therapy relies on a system that delivers all the necessary tools to bake the cake. The summary of the recipe that we know is called transgene. A key-operated switch to turn the oven on, known as promoter. A thermostat to maintain the desired temperature inside the oven, known as enhancer. The kit allows our cells to bake a cake that, although it's not the original one, it is still functional. Sometimes, if the original protein or cake was too big, the kit will allow our cells to bake a cupcake, a smaller version of the protein, but still fully functional. Gene therapy is advancing rapidly, and it has the potential to revolutionise healthcare and transform the lives of people with serious and life-threatening conditions.